brought us pizza and a wonderful presentation. So we have Ellen, who's going to give us a really great presentation on protein structure.
You guys hear me? So as your teacher said, my name is Ellen Lawrence. I work at Emergent BioSolutions. I'm a principal scientist there in the analytical development department, and primarily do assay development and run testing. So today we're going to talk a little bit about proteins. I'll give you a little bit about me, my background, education, and work experience. Go through some of the basics on protein structure. When you talk about protein structure, you always have to address amino acids. We'll talk about different ways that the structures of proteins are determined, and then get into the testing applications that we use pretty much every day. So a little bit about me. A long time ago, I was where you guys are in high school. I finished high school way back in 1993. I went to SUNY Portland and got my bachelor's degree there, so I majored in biology and started working full-time right after I graduated. I completed my master's degrees part-time, so I have an MS in Biotechnology and an MBA from Hopkins, and I did those part-time while I was working full-time. First job out of college was at Harvard Medical School. I was out there for a couple of months doing research on chronic myeloid leukemia. Spent a couple of years at Syracuse University doing research on a little protein called a calcium sensing receptor. Then made the leap to industry, worked at Bristol Myers Squibb for a couple of years, relocated down here to Maryland because biotech is a lot more prominent as a job opportunity down here. Worked at a small startup called Lentigen for a few years before making the leap to Emergent. I've been in Emergent since February of 2009. So, the real thing we want to talk about, proteins. What are proteins? They are large macromolecules consisting of amino acid residues. They are extremely important in the functions in cells. They are essentially the workhorses of cells. Human cell, each human cell is estimated to contain between 1 and 3 billion proteins. And these proteins participate in virtually every process within a living cell. So what do? They have several different roles. They are enzymes. They catalyze chemical reactions, things like DNA transcription, DNA repair, translation. There are over 4,000 reactions that are known to be catalyzed by enzymes. They're involved in cell signaling and signal transduction. So you have a lot of proteins that sit on the surface of your cells that when activated, they start a cascade of signaling throughout the cells. Things like insulin, membrane proteins. Ligand binding, they alter the permeability of the cell membrane, so a lot of charged molecules, large molecules can't get across the membrane by diffusion alone, they need help. So different proteins form channels to help these different molecules or ions across. They give us our immunity. We have 5 different types of antibodies within our bodies that help us to fend off different invaders like viruses and bacteria. And then they're structural proteins. So things like hair, fingernails, that kind of stuff, those are made up of proteins. The proteins confer stiffness, rigidity, and they also perform structural functions, so there's also some motor proteins. When you get into protein structure, it's kind of complicated. There's 4 different levels of protein structure. The building blocks are amino acids. When you talk about the primary structure of a protein, you're talking about just the sequence, the linear sequence of amino acids that have been combined to build the protein. They have a C-terminus end, a carboxyl end, and an amino end, an N-terminus. And in our cells, proteins are synthesized from the N-terminus to the C-terminus. When you look at primary structure, you look at amino acids, what are they? They're the building blocks. They're very important. They're pretty cool little structures. They're organic compounds. Every single one of them has an amine group. Every single one of them has a carboxyl group. Those are at opposite ends of the molecule. They have a central carbon with 1 hydrogen atom attached, and then they have an attached side chain. And what differentiates the amino acids from each other is the side chain that's attached to each and every one of them. The individual amino acids get linked by a special type of bond called a peptide bond. Each linked amino acid within a protein is then called an amino acid residue. The carbon, nitrogen, and oxygen atoms that make up the amino acid form the main chain or the backbone of the protein. And then the peptide bonds that form in between the amino acids, and that's through a reaction called dehydration synthesis. So what happens is every time a peptide bond is formed, you also get a molecule of water that is created. There are 22 natural or proteinogenic amino acids. There are a lot of others that exist, but they are not built into proteins, so they're non-proteinogenic. 20 out of the 22 amino acids are encoded by the universal genetic code. Those are known as standard amino acids. Of those, 9 are essential, meaning you can't produce them in your body. You have to get them through nutrition. You have to get them through what you're eating. 6 are conditionally essential, meaning that under certain pathophysiological conditions, we can't make them. And then 5 are dispensable, meaning we can synthesize them in our bodies at any time that we need them. There are 2 manufactured by special synthetic mechanisms. Selenocysteine is an amino acid that if our mRNA is coding with a specific sequence, we can take cysteine and alter it to make selenocysteine. The other is pyrrolysine. We don't use that in our bodies. It's used by some methanegetic archaea bacteria. So how do you code for the amino acids that you're building into a protein? The primary structure, the amino acid sequence, all encoded by our DNA, so it's all in our genes. The genetic code contains 3 nucleotide sets called codons, and each codon codes specifically for an amino acid. And when you look at the genetic code, you can kind of see it represented a couple different ways. Sometimes it's in this chart format where you can look at the first, second, third letter, see what amino acid it codes for. Other times it's this wheel representation where you start at the center, work outward, and at the very edge it tells you the amino acid coded for by the 3 nucleotide sequence. So building on the primary structure, once you get the amino acids linearly linked, you get these things called secondary structures, which are just repeating local structures. They are stabilized by hydrogen bonds that form between the carbon, nitrogen, and oxygen atoms of the backbone. Most common are alpha helices, beta sheets, and beta turns. Occasionally you'll get other things like kinks and whatnot. Those are non-common, and the protein will be degraded and destroyed. Secondary structure builds into tertiary structure. This is the overall 3-dimensional shape of a single protein molecule. It is kind of the spatial relationship of those secondary structures and how they are oriented alongside one another. These are generally stabilized by non-local interactions, meaning things like hydrophobic cores. Additionally, you can get salt bridges, hydrogen bonding, disulfide bonds, and post-translational modifications. This structure is extremely important though because that overall 3-dimensional shape of the protein actually controls its functionality. And this is just an illustration of some of the different types of interactions that occur, hydrogen bonding, disulfide bridges between the cysteines, that kind of stuff. And then the final level of structure is the quaternary structure. This is something that's formed when you have several protein molecules, so several polypeptide chains that interact with one another. They kind of like combine, but they function only as like a single molecule. So you have kind of several things intertwined doing one function. This again is a very 3-dimensional structure. It's formed by hydrogen bonding, covalent bonding. You can get hydrophobic packing and hydrophilic exposure. So the exposure to water, and we're largely aqueous, our bodies, does a lot to determine the overall structure of these proteins. So how do we determine the structure? We know it's very complex. We know we have all these different levels of structure. We need to know how do we determine the shape. There's a bunch of different ways. You can do X-ray crystallography. You take crystallized protein, you subject it to X-rays, and you can get down to atom structure of a protein. You can use nuclear magnetic resonance spectroscopy. This will give you kind of the structural information that you can get from X-ray crystallography. It also gives you the estimates of distances between different atoms within your protein. There is a quantitative method called dual polarization interferometry that measures the overall shape, which is called the conformation. So the overall conformation of the protein, and then you can also measure changes. So if you activate a protein, you can use this to kind of measure how does that shape change. Cryo-electron microscopy gives you lower level structural information about different protein complexes, and then electron crystallography gives you high resolution information, particularly for 2-dimensional crystals of membrane proteins, and membrane proteins are really, really difficult to crystallize. So we take all this information and we apply it for testing. And what we're testing are the products that we're making, typically recombinant proteins that go into a vaccine or possibly antibodies that are used for a biotherapeutic. We use SDS-PAGE and IEF, which are two different types of gel electrophoresis. We want to know the concentration of our proteins, how much are we making when we're running our manufacturing process. Western blotting is a way to kind of specifically identify whether the protein we're making is what we think it is, and then bioassays. So we'll start with SDS-PAGE. SDS is just an acronym for sodium bodecyl sulfate. That it's an anionic detergent, and what it does is it kind of equilibrates the charge among proteins so that you can then distribute them or separate them by size. So the SDS imparts a negative charge to evenly distribute the charge per unit mass of protein. You then load the samples onto a gel, you apply an electric field across the gel. Because the proteins are now negatively charged, they're going to move away from the negative pole toward the positive pole. And as they're navigating through this matrix of polyacrylamide, they're moving through pores. So the smaller proteins can move further because they move more easily. The larger proteins tend to get hung up more, they stay higher up in the gel. We run denatured versus non-denatured, meaning denatured just means we're linearizing the protein more. We're going in and treating it with either dithiothiazole, DTT, or beta-mercaptoethanol, and that breaks disulfide linkages that happen within the tertiary structure of the protein, and it also unfolds a little bit the quaternary structure. And we always run against a molecular weight ladder that consists of proteins of known molecular weights, so that when we see our sample, we can estimate the size of the protein and know, is it the expected size? So this is just an example of one of the gels that we've run throughout the years that I've been over at Emergent. And this is just an illustration of the type of apparatus that's used. So you use a chamber that has kind of an inner buffer core and an outer buffer core. And that's what establishes your anode and your cathode, your positive and your negative poles. The second type of gel electrophoresis we run, it's called IEF. It's called isoelectric focusing. And what it does is it separates proteins by the differences in their isoelectric point. Isoelectric point is just the pH at which a molecule doesn't have any net charge. So it's either taken on or lost different hydrogen atoms so that it's neutral. Samples in this type of technique, they're added to a gel that has an immobilized pH gradient, and you can actually order the range of pH that you're looking to test for. The samples are again analyzed against a PI marker so that we know, what part of the gel represents a certain PI. Well, an electric field is applied toward the gel. Negative molecules go toward the positive end of the gel, the anode. Positive molecules go toward the negative end of the gel, the cathode. Molecules will migrate until they hit that point in the gel where their pH, the pH confers a net charge of 0, so their isoelectric point. And this is a good way, if you have two proteins that are the same size, it's a good way of distinguishing between the proteins because they're not going to have the same PI. You can take IEF and SDS-PAGE and put them together for 2D gel electrophoresis. You first separate proteins by their PI, so by their charge, then by their size. So you add your sample to this special little gel strip, apply an electric field, the proteins will move along the strip until they hit their PI. Then you take that strip, you load it into a gel that has one very large sample well, and then you apply the electric field and you separate the proteins based on their size after they've been separated by charge. And what you get is this big scattering of proteins. So every dot on that particular gel represents an individual protein. So you can go in and look at specific proteins. So protein concentration, how much of our protein do we have? We have a couple techniques that we use for that. The first is A280. That's just absorbance at a wavelength of 280 nanometers. We do this with spectrophotometers. The relationship between absorbance and protein concentration is linear. It is directly proportional. The more protein you have in solution, the higher your absorbance is going to be. Proteins absorb light at two different wavelengths. They'll absorb at 200 nanometers due to the peptide bonds in the protein, and then they'll absorb at 280 nanometers due to absorbance primarily by amino acids with aromatic rings. The two that are more responsible for that are tyrosine and tryptophan. And then your disulfide bonds also are somewhat responsible for your absorbance at 280. You can take your absorbance and plug it into an equation. It's called the Beer-Lambert law. says absorbance is your extinction coefficient, your path length, which is always known based on the cuvette that you're using for your absorbance reading. And then you can solve for concentration. So you can ultimately determine how much of your protein you have. BCA, bisinkininic acid assay, is the other way to measure protein concentration. This gives you total protein. It's a ion reducing assay. So it reduces carbon 2 ions from copper 2 sulfate, copper 2 ions from copper 2 sulfate to copper 1 ions. 2 molecules of BCA chelate with each copper 1 ion and you get a purple color. And then you can measure the absorbance of the purple color at a wavelength of 562 nanometers. Your color change is directly proportional to the amount of protein present. So the more protein you have, the darker the purple color is going to be. And this is because the amount of carbon 2 reduced is directly proportional to the amount of protein present. Western blotting builds on SDS page. You have to run an SDS page gel before you can do a blot. It is a way to detect a specific protein. So here you're looking for a specific protein. You want to know, you run your manufacturing process, are you making what you think you're making? You run the gel, the SDS page gel. You then arrange the gel between filter papers and a membrane, either a PVDF or a nitrocellulose membrane. You again apply an electric field. The proteins migrate from the gel onto the membrane. The membrane is then probed with an antibody that is specific for the protein that you're looking for. You wash off any excess reagents, probe the membrane again with a secondary antibody, which looks for the first antibody to stuck to your membrane. And your secondary antibody is going to be conjugated to some type of either an enzyme, a fluorophore, or a radioisotope. So membranes that are conjugated to enzyme, you can do colorimetric or chemiluminescent detection, meaning it's either a color change or it produces light. Color changes, you dry the membrane, scan it with a densitometer. For chemiluminescent, you can actually take your membrane and press it against a piece of film in the darkroom, develop your film, and you get little dark bands wherever you have antigen and antibodies that stuck on the membrane. And lastly, we run bioassays. We run actually a lot of bioassays. You can have cell-based bioassays or non-cell-based. I'll give you an example. One that is cell-based is called ADCC. It's antibody-dependent cell-mediated cytotoxicity. And here you're essentially using immune cells that have been genetically engineered to produce a substrate that when you apply an enzyme, it'll produce light. So you take target cells that express your antigen. You add in your antibody. This is typically used with IgG antibodies. Your antibody sticks to your target cells. You then take that, mix it with your immune cells that have been genetically engineered, and after a little while, you add in your enzyme, and if there's antibody there that's recognizing the antigen, you get light produced, which you can measure with the luminometer. So the idea is the more functional antibody you have in a sample, the more light you would get as an output. And this is really, really important. FDA now really wants us looking at the activation of certain receptors by the antibodies that we're making. And then non-cell based, the best known of which is ELISA, enzyme-linked immunosorbent assay. This is commonly used for protein detection and quantitation. We use it a lot. I don't think we've had a project that's come through Emergent where we haven't used ELISA in one way or another. These are very specific, they're very robust, they're very repeatable. The results are generated by an absorbance reading, and that is from a substrate color change. So you mobilize your antigen on 96 well plastic plates, you add in your antibody, you add in a conjugated secondary antibody that's against your antibody that you're trying to detect, and then add substrate and you get color change. And in between each of these steps, you do wash steps to wash away any excess reagents. But ultimately, again, kind of like your BCA, kind of like your ADCC, the amount of color change or light, however you're detecting your antigen, is directly proportional to how much antibody you have. So if you're getting more color change, then there's more of your product there. If you're looking for an antigen, typically you have to use a different type of ELISA. It's called a capture ELISA or a sandwich ELISA. And this is where you're mobilizing an antibody that recognizes your product on plates you add in your product, and then you have to add a detection antibody followed by another effect a color change. So that's a lot of information in a very short period of time. I'm happy to answer any questions that you ask.
What do you typically study proteins for?
We use them in vaccines, so we use them to stimulate immunity so that you'll recognize, for example, anthrax if you were to be infected with anthrax. You can also use them as biotherapeutics. Those are typically more the IgG antibodies. So you can provide a patient with an IgG antibody that will tangle up, either a bacteria or a virus if that's the case. Some of them are used for autoimmune diseases, but they're used either in vaccines or as therapeutics.
So they're just mainly used for immune systems.
Well, it's either to, in the case of a vaccine, you're stimulating your immune system so that you'll recognize either a virus or a bacteria if it infects you. And in other cases where you're doing like an IgG antibody, you could be treating something somebody's already been infected with, or you could be treating something like cancer, you could be treating, like I said, an autoimmune disease. So if there's a particular protein that's being broken down by your body, where your body is kind of attacking itself, the antibody will stop that. Other questions?
When you're using that protein detecting method that you were talking about, how do you know when you found like the protein that you're looking for? Is it darker than yours?
Which one, the 2D gel?
Yeah.
Usually you have a pretty good idea of size and charge, and what you're really trying to do is isolate it from the gel. So you're looking to see, okay, we're expecting it to be this particular size, this particular charge. Are we getting anything in that area? Sometimes it's darker, sometimes it's faint. It just depends on how much you're looking. Other questions?
Not exactly, yes. How can your work relate to helping people who have Ig deficiencies?
We can treat them. We can give them antibodies that will, detect and attack what they're unable to do on their own. So you can do courses of antibody therapy, and that will basically enhance their immune system and give them the immunity that they are unable to produce on their own.
You said that we have 9 antibodies that we produce from our own body with the nutritions that we have.
There are 9 essential amino acids that we can't produce. So we have 5 that we can.
Oh, okay. So what happens if you produce too much, or what are the consequences?
Your body will essentially regulate itself. So if, let's say, you're eating a bunch of food and you're getting a particular essential amino acid, you'll use what you need and then your body excretes the rest. It just goes out through your natural waste systems. Other questions?
So you said that some of the amino acids we get are from nutrients, and I guess the resources we have in different parts of the world are different. Is it possible to live in a place where you can't get a certain amino acid because it's not present in your diet?
I mean, it's kind of highly unlikely because of the way that the flora around the world is distributed. You can usually get what you need either from a source of meat or some type of vegetable. And the vegetables that are common kind of throughout the world tend to have the majority of what you need. Otherwise, you can always take a supplement. You can take different types of vitamins that will also contain amino acids.
In your line of work each day, would you say that like you try to find a vaccine for disease like by using the protein finding methods like
Primarily what I do is test the proteins that have already been found. We're not really into
Log me out.